Welcome to the Deep Dive. Today we are taking a fascinating plunge into the world of plant biology, specifically tackling a question that seems simple on the surface. How do plants know when to flower?
It sounds straightforward, but it's one of the most critical decisions a plant makes. I mean, get it wrong, flower too early, maybe hit a late frost, or too late and miss your pollinators.
Right.
And you've basically lost your chance to reproduce that season. Timing is absolutely everything.
And understanding this precise timing mechanism, that's our mission today. We're pulling insights straight from a key chapter in Plant Physiology and Development, 6th edition, to really unpack the environmental cues and the molecular signals involved.
Yeah, we want to trace the whole journey, How do plants keep track of seasons, even the time of day? What environmental signals actually matter, and how are they even detected? And then how do those signals trigger the big internal change that leads to flowering?
It all builds towards something the source calls floral evocation, which is kind of the plant's point of no return, isn't it?
Exactly. It's that moment the growing tip commits, decides to make flowers instead of just, more leaves. It's a fundamental developmental switch.
Okay, let's dive in and see how they pull it off.
Right.
So before a plant can even consider flowering, it has to, grow up first,
Yeah.
right? It goes through distinct life stages, a bit like us going from infancy to adulthood.
That's a good way to put it. After a seed sprouts, a plant enters these post-embryonic phases. There's typically a juvenile phase, then the adult vegetative phase, and finally the adult reproductive phase.
And the shift between them is called phase change.
Correct. And the really crucial bit is that transition to the adult vegetative phase, because that's when the plant actually gains what they call the competence to flower.
Ah, so it can flower now, even if it still needs other signals to actually do it.
Precisely. It's now receptive. And you can often spot this phase change just by looking. English ivy is the classic example they use.
Oh, yeah.
Yeah, the leaves lower down. From the juvenile part, they're lobed, kind of star-shaped. But the leaves higher up, on the adult shoots that will eventually flower, they're completely different ovate, simpler shape, arranged differently. You can literally see this gradient.
I've definitely seen that on ivy. So the older tissue at the base is juvenile, and the newer stuff at the top is adult.
Exactly that. And the time it takes to hit this adult phase, well, it varies massively. Some little annuals are ready in days.
Wow.
But then you have big trees like oaks that might take decades.
Decades. Is it just about age then, or is something else going on?
The source suggests plant size or maybe its developmental stage is often more critical than just how many years have passed.
Okay.
Things that slow growth, like low light or water stress, they can actually keep a plant juvenile for longer.
Makes sense.
Whereas, give a plant great conditions, vigorous growth, and it tends to mature faster. Sometimes it's about reaching a certain number of leaves.
So they need to build up enough resources, enough structure before they're ready to reproduce.
That seems to be a big part of it. And once they do hit that adult phase, it's pretty stable. Like, if you take cuttings from the juvenile base of that ivy, they grow as juvenile plants.
Right.
Cuttings from the adult tips stay adult. The state is kind of locked into the tissue.
That grafting example was really neat too. You mentioned grafting juvenile herbaceous plants onto a flowering adult can sometimes kickstart flowering.
Yeah, it's like the signal is there floating around from the adult part, and the juvenile bit just needs to be receptive enough to hear it. But interestingly, juvenile woody plants often don't respond that way.
Ah, so they truly lack that competence. They're just not ready internally.
Exactly. Shows it's not quite the same mechanism for all plants. As for the signal's driving phase change, carbohydrate status seems important. Low light means low sugars, like sucrose, and that can prolong juvenility. There's a molecule mentioned, trehalose 6-phosphate, which seems to link sugar levels directly to flowering pathways in Arabidopsis. And plant hormones, specifically gibberellins, can induce flowering in young conifers, suggesting they push towards reproduction.
And then it gets really small scale, down to tiny RNA molecules. You're saying they're involved in this whole growing up process.
Absolutely central. There's a microRNA called miR-156. It's a major player in the juvenile to adult transition in loads of plants.
miR-156.
Yeah, think of high levels of miR-156 as basically putting the brakes on, keeping the plant juvenile. As the plant develops over time, miR-156 levels gradually drop.
And when miR-156 levels fall, what kicks in.
That drop allows certain target genes called SPL genes to become active, and these SPL genes are the ones that promote the adult characteristics.
So the decrease in miR-156 is like slowly releasing the handbrake.
That's a perfect analogy, and it's not alone. There's another microRNA, miR-172, that worked kind of in tandem. As miR-156 goes down, miR-172 levels tend to go up. And miR-172 is also influenced by things like day length, and it targets genes that normally repress flowering, genes called AP2-like proteins.
Wait, hang on. So miR-172 actively shuts down the genes that are telling the plant not to flower.
You got it. By knocking out those repressors, miR-172 helps clear the path for the plant to actually enter the adult reproductive phase. It's this really intricate dance between these tiny RNAs sensing internal state and external cues that controls developmental readiness.
Wow. Okay, so the plant's grown up, it's competent. Now it needs to know when the time is right. And that brings us to the plant's internal sense of time circadian rhythms.
Yep. Just like us, plants have an internal clock. It anticipates the daily cycles of light and dark, governs all sorts of things, leaf movements, when pores open, metabolic timing.
And this clock keeps ticking, even if you put the plant in constant light or constant darkness, right? Shows it's driven internally.
Exactly, by an endogenous oscillator. The natural period of this clock is usually close to 24 hours, hence circadian, around a day. But because it's not exactly 24 hours, it needs to be reset or entrained every day by environmental signals. They call these Zeitgebers time givers.
Like sunrise and sunset.
Primarily, yeah. Dawn and dusk are the main ones. These light signals synchronize the internal clock to the actual 24-hour day. Without them, the rhythm just free runs at its own natural pace.
And this clock is surprisingly stable, isn't it? Like, temperature changes don't mess it up too much.
That's a key feature, temperature compensation. Really important for keeping accurate time when the weather's changing. It can also adjust to different day length through phase shifting.
Phase shifting.
Yeah, like if you flash a light early in the plant's subjective night when its internal clock thinks it should be dark, it can delay the whole rhythm. A flash late in the subjective night advances it. This helps the plant stay synced up as the seasons and day lengths change.
Okay, so what's actually seeing the light to reset the clock? Which sensors?
It's the usual photoreceptor suspects, phytochromes, which see red and far-red light, and cryptochromes, which see blue light. Studies in the model plant Arabidopsis show multiple phytochromes and cryptochromes, CRY1 and CRY2, are involved. There's even evidence that the cryptochromes might help relay phytochrome signals to the clock mechanism. And remember, CRY2. Activated CRY2 can also directly turn on a key flowering gene we'll get to later.
Wow, so it's linking light perception directly to the flowering command pathway.
Exactly. Okay, so the plant has this amazing internal clock. How does it use that clock to figure out the season? Because, different seasons mean different day lengths.
Right, that's where photoperiodism comes in, measuring the day length.
Precisely. It's the ability to use day length as a really reliable calendar signal. Day lengths changes predictably depending on your latitude and the time of year.
And the classic experiment here, you have to mention it, is the Maryland mammoth tobacco back in the 1920s.
Oh, absolutely. Foundational work by Garner and Allard. They had this mutant tobacco variety that just grew huge but wouldn't flower in the summer fields in Maryland. They figured out it wasn't about, say, accumulating enough food during long summer days. It was specifically about the length of the day, or as it turned out, the night. If they covered the plants part of the day to shorten the light exposure, boom, they flowered.
Which led to classifying plants based on their day length preferences for flowering.
Yep. You've got your short day plants, SDPs, which flower when days are short, technically when the day is shorter than some critical length, and long day plants, LDPs, which flower when days are long, longer than their critical length.
And some are strict about it, qualitative, while others just flower faster with the right day length, quantitative.
Exactly. And then there are those really interesting dual day length plants that need a specific sequence, like long days, then short days, or vice versa.
And we can't forget the day neutral plants, the DNPs.
Right. Super important. They just flower when they're ready, pretty much regardless of day length. Their timing is mainly under internal or autonomous control. Think kidney beans, lots of desert annuals.
And experiments showed it's the leaf that perceives this day length signal.
Yes, that was another key finding. Classic experiments with xanthium and SDP. They induced just one single leaf with short days, kept the rest of the plant in long days, and the whole plant flowered.
Wow.
And grafting studies, like with perilla, also confirmed it. You graft an induced leaf onto a non-induced plant, and the recipient flowers. A signal clearly moves from the leaf. This perception in the leaf is called photoperiodic induction.
Okay, now prepare yourselves because here's the really counterintuitive bit from the source material. It turns out plants aren't really measuring the length of the day.
Right. This is the crucial insight. They are primarily measuring the duration of the night.
The darkness.
Yes. So SDPs actually need a continuous period of darkness that's longer than some critical night length. LDPs need a night that's shorter than their critical night length.
And the proof is the famous night break experiment.
Exactly. Take an SDP, put it in conditions with a long night that should make it flower, then interrupt that long night, even with just a brief flash of light. And it prevents flowering. It's like you reset the plant's night timer. But interrupting a long day with a period of darkness doesn't matter.
That's just wild. So even a few minutes of light splashing onto an SDP in the middle of its required long night stops it flowering.
That's the power of the night break. And the timing of that flash is critical. It's most effective right around the middle of the dark period. For both SDPs and LDPs, interrupting the middle of the night has the biggest effect.
Which points straight back to the circadian clock, doesn't it? That sensitivity changes over the night.
Precisely. It supports the clock hypothesis. They use their internal circadian oscillator to measure the duration of the night. Experiments with soybean and SDP in continuous darkness show that its sensitivity to a night wind cycled with circadian rhythm.
So it's not just about having a night that's long enough. It's about when dawn arrives or when a light flash hits relative to a specific phase of the internal clock.
That's the essence of the coincidence model. Flowering is triggered when light exposure coincides with a particular light-sensitive phase dictated by the circadian rhythm. Okay. For SDPs, light hitting during that sensitive phase inhibits flowering. For LDPs, light during that sensitive phase promotes it. And that sensitive phase keeps oscillating even if the plant stays in the dark.
This is getting beautifully complex. Okay, how do these light signals and the clock actually translate into a molecular decision to flower? Let's get into the genes and proteins.
Right. Let's start with long-day plants, like Arabidopsis. A really key gene here is called constans, usually just called CUTO.
Constans, okay.
CO produces a protein, a transcription factor that regulates other genes. Now, the CO gene gets switched on and off by the circadian clock. Its mRNA levels cycle, usually peaking about 12 hours or so after dawn.
Okay, clock controls the gene expression, but you mentioned the link to light earlier.
Yes. Here's the clever part, the coincidence mechanism for LDPs. The CO protein itself is unstable. It gets broken down really quickly in the dark. But light, specifically blue and far red light sensed by cryptochromes and phytochromes, stabilizes the CO protein.
So in long days, the clock makes the CO mRNA peak happen when it's still light out.
Exactly. So the protein gets made, it's stabilized by the light, it accumulates.
And it promotes flowering.
Right. But in short days, that mRNA peak happens when it's already dark. The protein gets made, but whoosh, it's immediately degraded. No accumulation, no flowering signal.
Brilliant. Okay, so CO protein builds up in the light when the clock gives the green light. What does the CO protein actually do?
It turns on the expression of another crucial gene. This one's called flowering locus T, or FT.
FT.
And FT, well, there's overwhelming evidence now that the FT protein is the long-sought, almost mythical floral signal florigen.
No way. They actually found florigen after all that searching.
It seems so. FT ticks all the boxes. It's expressed in the phloem companion cells and the leaves, right where the light signal and CO are doing their thing. The FT protein itself is small, it's mobile, and it moves through the phloem, the plant's vascular highway, all the way up to the shoot apical meristem, the growing tip. And when it gets there, it triggers floral evocation. It is the mobile signal.
That is incredible. Okay, what about short-day plants, like rice? Is it a totally different system?
Amazingly, no. They used very similar machinery, homologous genes, basically, the rice versions of CO and FT.
Really?
Yeah. In rice, the CO equivalent is called heading date 1, HD1, and the FT equivalent is heading date 3A, HD3A. And just like CO, the HD1 mRNA levels cycle with the clock peaking around the same time.
So same genes, same clock timing,
Yeah.
but they need short days. How does that work?
Here's the twist. In rice and SDP, the HD1 protein acts as a repressor of the FT3A and SD3A.
A repressor. So CO promotes FT in LDPs, but HD1 inhibits HD3A in SDPs.
In the light, yes. The coincidence of HD1 expression in light actually inhibits flowering in rice by blocking HD3A production.
Okay, my head's spinning slightly. So for rice to flower, the HD1 protein needs to be there, driven by the clock, but needs to be there when it's dark.
Precisely. When HD1 is expressed in the dark, it doesn't repress HD3A. So HD3A gets made, the FT-like signal is produced, moves to the apex, and the rice plant flowers. It only happens on short days when the HD1 peak occurs during the long night.
That is remarkable. The same core components, just wired differently, promotion versus repression in the light to get opposite photoperiod responses.
Isn't it elegant? It shows how evolution can tinker with existing pathways.
We touched on photoreceptors for the clock, but they're also directly sensing the light for the flowering response itself, like in the night break.
Absolutely. Phytochromes are central, those night break experiments. Interrupting the night in an SDP with red light stops flowering. But if you follow that immediately with far red light...
It reverses the effect. Classic phytochrome.
Textbook phytochrome action, yeah.
Yeah.
And action spectra plotting effectiveness against wavelength confirm that the light that affects flowering matches phytochrome's absorption peaks. Plus, mutations messing up phytochromes often cause plants to flower at weird times, ignoring day length.
And the blue light receptors, cryptochromes.
Also important, especially in LDPs like Arabidopsis. Remember we said they help stabilize CO protein? Well, mutations in the CRY2 cryptochrome delay flowering specifically under long days, so they're definitely part of perceiving that long day signal and linking it via CO stability to the FT output.
Okay, so light, the clock, day length. That's a huge part of the timing puzzle. But there's another major environmental cue, especially for plants in temperate climates. Cold.
Vernalization.
Yes, vernalization. It's the promotion of flowering by exposing the plant to a prolonged period of cold, typically just above freezing, say 1 to 7 degrees Celsius.
And it has to be a wet seed or a growing plant, right? Not just a dry seed in the fridge.
Correct. It requires active metabolism. The plant needs to be hydrated and physiologically active, even if growth is slow in the cold. Without this cold treatment, many plants think winter wheat or biennials like carrots or cabbage would just stay vegetative or flower very late.
So the cold is basically telling them, okay, winter has happened, you've survived, now you can think about flowering when spring comes. That's exactly the ecological logic. It prevents them from flowering in, say, a warm spell in autumn, only to be killed by winter frost. The cold requirement ensures they overwinter first. It usually takes several weeks of cold, and the longer the cold, generally the stronger and more stable the effect. It gets harder to devernalize with subsequent warmth.
Where does the plant actually sense this cold signal? Is it the leaves again?
No, this time it's primarily the shoot apical meristem, that crucial growing tip where the decision to flower is made. Experiments chilling just the apex can induce flowering. Vernalization makes the meristem competent to respond to other signals, like the long days of spring that often follow winter.
Cold first, then maybe long days. Okay. But how does the plant remember it was cold? Weeks or months later, when conditions are good, how does it know it fulfilled its cold requirement? That sounds like memory.
It absolutely is a form of biological memory, and the mechanism involves epigenetic changes.
Epigenetics. So changes in gene expression that don't involve changing the actual DNA code.
Exactly. Stable alterations in how genes are read. In Arabidopsis winter annuals, there's a powerful flowering repressor gene called flowering locus C, or FLC.
FLC, okay.
Before vernalization, FLC is highly expressed. It's like a big stop high sign preventing flowering.
And the cold knocks down the stop sign.
It essentially switches FLC off epigenetically. The prolonged cold triggers modifications to the chromatin, the packaging of DNA, with histone proteins around the FLC gene.
Ah, changing the packaging.
Right. It shifts the chromatin from an open, active state to a closed, inactive state. This silences FLC expression, specifically in the cells of the meristem that will form the flower.
So the cold doesn't delete the FLC gene, it just locks it away, makes it unreadable.
That's a great way to think about it. And this silent state is remarkably stable. It's maintained through cell divisions as the plant grows, even long after the cold is gone, so the plant remembers winter.
And that allows flowering when other conditions like long days triggering FT are finally met.
Precisely. Because FLC normally works by repressing FT and other flowering promoter genes. Once FLC is silenced by cold, the path is clear for FT to work when the photoperiod signal arrives.
Is this FLC system universal for all plants that need vernalization?
It seems not entirely. In cereals like wheat and barley, a different key repressor called VRN2 seems central to their vernalization response. It suggests that the ability to use winter cold as a cue might have evolved independently in different plant lineages.
Interesting. And they still need to figure out how plants measure the duration of cold, right, to distinguish winter from just a cold snap.
Yeah, that's still an active research area. How do they integrate temperature over weeks?
Okay, so we have leaves sensing light via the clock, the apex sensing cold via epigenetics, but the flowering happens at the apex. That signal from the leaf, phlorogen, has to travel.
Yes, the photoperiod signal especially. The evidence for a mobile signal goes way back to those grafting experiments in the 1930s by Mikhail Chilakian. He's the one who coined the term phlorogen.
Right, grafting a leaf that's seen the right day length onto a plant that hasn't, and poof, the recipient flowers.
Exactly. Classic experiment.
Hmm.
Graft one induced leaf from a perilla plant, short day treated, onto a non-induced plant, kept in long days, and the whole non-induced plant flowers.
Amazing.
And what was really striking was that this worked even between plants with different photoperiod requirements, like grafting an SDP leaf onto an LDP, sometimes even between different genera.
Wow, suggesting it might be a universal or at least highly conserved signal.
That was the strong implication. And further studies showed the signal moved in the phloem, along with sugars. If you block phloem transport, you block flowering induction. It traveled at about the same speed as sugar translocation.
Which brings us neatly back full circle to FT. You said the FT protein is phlorogen.
That's the major synthesis that emerged from molecular genetics. Decades of searching for the chemical nature of phlorogen were tough, but genetics cracked it. Experiments showed that CO, the protein upstream in the leaf, acts in the phloem cells. And its key downstream target, made in those same phloem cells, is FT. Introducing the FT gene directly into plants often causes them to flower like crazy, regardless of day length.
Fulfilling the role of a powerful floral promoter.
Exactly. And crucially, researchers demonstrated that the FT protein itself can physically move from the leaves through the phloem up to the shoot apical meristem. Its movement and its function perfectly match the predicted properties of phlorogen.
So FT protein, euphlorogen, the mobile command signal.
That's the current consensus, yeah. It beautifully integrates all those decades of physiological work with the molecular details. The source also mentions hormones like gibberellins and ethylene can influence flowering, but FT is considered the primary long-distance photoperiod signal.
Well, this has been quite the deep dive. We've gone from whole plant behavior right down to microRNAs and epigenetic marks. It's clear that deciding when to flower is incredibly complex for a plant.
It really is. So much more than just waiting for warm weather. It's this sophisticated integration of internal readiness, age, size, competence, with really precise environmental tracking.
Using day length via photoperiodism, and for many, winter cold via vernalization.
And employing those internal circadian clocks to measure night length with remarkable accuracy, using phytochromes and cryptochromes to actually see the light signals.
All feeding into these molecular pathways with key players like CO controlling stability, FLC acting as an epigenetic brake, and FT being the final trigger.
And that final command, phlorogen, turning out to be this mobile FT protein produced in the leaves based on all those inputs and traveling up to the apex to say, okay, time to switch.
Understanding this isn't just, cool biology. It's vital for agriculture, breeding crops that flower at the right time in different climates, or manipulating flowering for better yields.
Absolutely. And it's crucial for understanding how plants might cope or struggle as climate change shifts these seasonal cues like temperature and potentially alters day-length perception indirectly.
I think we've covered the key ground from that chapter. The developmental stages like phase change, the timekeeping with circadian rhythms, sensing light with photoperiodism and night breaks, the cold requirement of vernalization, the molecular nuts and bolts with CO, FLC, FT, and finally, identifying phlorogen.
Yeah, it provides a really solid framework for how plants manage this critical life transition.
Okay, here's something to chew on until our next deep dive. We've seen that plants have evolved all these different ways to sense their environment. Long day, short day, needing cold, not needing cold. Really diverse strategies. Yet they seem to converge on using a remarkably similar protein FT or its relatives as that final mobile flower now signal.
The universal phlorogen concept seems to hold up pretty well at the FT level.
So what does that tell us about evolution? Could it be that this FT signaling module is incredibly ancient, a fundamental switch for making flowers, and all the complex environmental sensing pathways? The photoperiodism, the vernalization evolved later as ways to control when that ancient FT button gets pressed in different lineages, adapting to different climates?
That's a fantastic question. It touches on deep ideas about evolutionary tinkering, conserved core pathways, and how complexity builds up around a fundamental process. A great thought to end on.
Thanks for joining us on the deep dive.
My pleasure. Always fascinating stuff.